It's all driven by techniques and the molecular biology techniques, as far as one can go back. It starts from 1977, where Sanger developed the sequencing technology. Since then, the technology was developing over the time, but there's a large gap of the technology development. But if you look at from 2005, it is going in a linear fashion, as you can see, not only from the sequencing technology, but also single cell technology, subcellular technology, mass spectrometry, multiplex IHC. We can keep going. So it's just kind of evolving in a larger way, and data is pouring in humongous way that it is now challenging how are we going to really put them together, analyze them, and then make sense out of it so that we can apply it wherever needed. As I mentioned earlier, the omics can be added to any part of the molecular biology, other than the, for example, radio biology, which is a CT scan or MRI that usually people take for any of the, so, that any of the for cancer screening or anything that people would take, and you can add also the pathology-based omics and so on and so forth, and that provides an idea probably for you to know that how much of data that can be generated, because recently people are going back to the CT scans and MRIs that were taken for decades, and then analyzing them in a lot of different ways, an unprecedented amount of information coming out of that, and it's quite exciting that how we can transform the lives of the patients. However, these technology and the omics data are not going to be completely useful unless otherwise we match that with the phenotype. What does phenotype mean? Phenotype means how long a patient survives, or if a patient would respond to a particular treatment or not, or the major aspects within any cancer patient that one would want to know. And that is what is called phenotype. However, those phenotype and information are not always readily available. In that case, we can use other information like the drug response data that are generated from the cell lines or from mouse models, some of the pathway analysis these days that can be done through bioinformatics. Can also play as a surrogate that through which you can really understand some of these patient phenotypic information. Maybe I just want to skip this. Basically, in this slide, I'm just not only either subtypes that we work, or we call, okay, these patients live longer, these patients do not live longer, like prognostic stratification, or a predictive stratification, which is like these patients would respond more to this therapy, these patients would not respond, and so on and so forth. But we can also integrate this with the phenotype, like, as I mentioned, the pathology, which is often readily available in the clinic with the H&E slides or other information. It can also be MRI and CT scans, but also the clinical information from the patients, which we call clinical covariates, like grade of the patient, stage 1, 2, 3, 4, kind of mutations they have, the treatment they have received, all can be integrated, which I will go through in later slides. There are different ways of integrating them to ask two major questions. Obviously, prognosis, survival of the patients, and the one is precise therapy. It means who to treat. So with precise or the cancer, I'm not going to go into the usual definition of what cancer means. It's highly proliferative cells, and those kind of usual definition of what cancer means. It's highly proliferative cells, and those kind of information. I just wanted to tell you that how complex cancer is. Cancer is highly heterogeneous. That means, as you can see, that each patient represents the cancer with different, even if it is colorectal, pancreatic, or breast cancer, within them, they come in a different flavors. But conventionally, it's being considered as breast cancer or colon cancer, and the treatments are provided in that patient's. Hence, it's very difficult to get treatments. In another way to say, one size doesn't fit all. So that means we need to think really in different modes. So that is what is personalized medicine. Personalized medicine, as I mentioned earlier as well, it is to match patients to therapies. That means instead of taking them as one single, cancer type, you can split them based on, for example, here I have put them by colors. But the colors here also mean which therapy to treat them with. Some patients probably do not need treatment. It is true, we have observed, but it is very difficult to implement in the clinic because majority, but it's quite important to also provide that. So how do we really decide? At this point, the decisions are made in a conventional way, but can we do that using multiomics? Can we use that using biomarkers? Can we use the MRI-based digital scanning that can provide this opportunity to do that? That's what is currently a great area to venture into, driving majority of the field, including this one. However, the complexity of the cancer doesn't stop there. It also has complexity within an individual cancer. For example, it is called intratumoral. When a patient's tumor differs from one patient to the other, it's called intertumoral response, which is what, intertumoral changes, which is what's shown on the right-hand side. Whereas can you see my arrow here?
Yes.
Hello?
Yes, Indra. Yeah.
So, all right. So, the intratumoral heterogeneity within an individual tumor can come in a different flavor. For example, I'm only considering here only the cancer cells. There could be multiple types of cancer cells that can be present here, which again is very complex because you try to target one cancer and then it can lead to other cancers to become resistant and come back again. So when you treat such kind of these heterogeneous cancers with the therapy, what happens is that some of them will respond. As you can see here, this particular patient probably had a complete response, which is great. But at the same time, some patients could become complicated, that it could metastasize to multiple organ, like in the blue tumor here. It all varies, depends on how much heterogeneity one tumor has within the individual patient. At the same time, it is also further complicated because cancer is not just cancer cells alone. It is also comprised of stromal and cancer cell interactions. There are different cell types. The most notable ones are cancer-associated fibroblasts, endothelial cells, pericytes. But this is like a colony of cells trapped among themselves. Sometimes they fight with each other. That means some of them are against cancer, some of them are for cancers. It doesn't matter, but these are all mutually interacting. And how they interact, how the tumor becomes highly heterogeneous. And most importantly, the immune cell plays a role, and that comes in a different immune cell types and flavors. And each of them be against the tumors and some of them for the tumor. And again, more heterogeneity exists because spatial heterogeneity you can call, for example, colorectal cancer can metastasize to different organs. Temporal heterogeneity, as I mentioned, in intensified therapy, patients receive first line, second line, third line, keep going, even fourth line. And of course, it depends on the patient and their health conditions. But each therapy we have to understand would influence these cancer cells, and they could change their forms and shapes and molecular characteristics and other things. So it is quite complicated, and here I'm just kind of summarizing some of these tumor heterogeneity, but you can keep going. And one of our interests is to see how we can take all the heterogeneity and phenotype from patient therapy response and organize into map. Yes, absolutely, it is highly ambitious. It doesn't matter whether you can achieve this or not in one's lifetime, but at least it's good to have such kind of larger vision. The map-like ecosystem probably you probably studying in your courses. And can we come up with such thing? Even if you can scratch the surface, that's a lot more for the patients and the clinicians. So how do we do that? Artificial intelligence and machine learning or deep learning methods and the data generated from the machine learning can come together, give a potential shape and provide an opportunity to do that. In the usual talks I give, how we have practiced one way or the other in colorectal cancer. But I'm just today going to give more introduction to artificial intelligence and how one could work on so, switch to artificial intelligence. Probably you know artificial intelligence is a larger umbrella term, which basically means that something can be done which mimics a human brain, but not done by the human, but done by the machine. It can go in any extent, whereas machine learning, deep learning comes within these umbrella subsets. Machine learning is the largest subset than the deep learning, where machine learning means that at one particular level, can machine learn and mimic human behavior. Whereas the deep learning is at the multiple level, deeper, can it go and learn and understand. So deep learning is much more complicated than the machine learning. But overall, artificial intelligence is beyond the. It's supposed to be beyond the humans can imagine. When you want once something which you can do in an Excel sheet doesn't require machine learning, unless otherwise you really want to do that. So artificial intelligence, it's a term, though it is a buzzword these days. It's existing since 1956. Probably most of us were not born. For example, Siri, that you probably use regularly, is an artificial intelligence. The self-driving car is also an artificial intelligence. There are other automated surgery training that is also available these days. I'm not going to go into the artificial intelligence much here. I'm going to talk more about machine learning and deep learning methods. So machine learning also comes in two flavors. Machine learning can be supervised or unsupervised. Supervised or unsupervised is in a way simple that when you have a set of balls, red, blue, green, whatever it is, and you just close your eyes and then you basically take each one of them and then put them into another box saying whether it's red or blue, completely unbiased, is unsupervised. But someone tell you a texture of how the red ball would look or a blue ball would look, and when you touch it in a simple way. There are different methods to really take a larger data and do unsupervised or supervised methods. I have only included just a couple of them, which most of you probably heard about, but there are also sophisticated ones. And we often use non-negative matrix factorization for unsupervised. We usually start with unsupervised and then go supervised, but you can also start with supervised, which I'm going to give a few examples. I just want to stop if there are any questions or anything, anything that you would like to ask. No questions. So the supervised and unsupervised methods can come in linear model or non-linear. Again, linear simply means that you can draw a simple line across, split them into two groups. Non-linear means you cannot really split them by a single line, then you have to really different ways of inferring that. Deep learning is while machine learning is, as I said, one layer. When you have one layer of understanding, it's called machine learning. When you have multiple layers, it's called deep learning. I will come to that, what these layers mean in a minute. Usually, any machine learning or deep learning would have an input layer and output layer. That means you can put in the balls, which I said red, blue, green, all mixed together as an input, and you're basically going to ask that each one of them represents red ball, green ball, and so on and so forth, is what the output you're expecting. And whatever the steps in between that you're going to do is what is the hidden layer. That basically when someone asks you to take the red ball, blue ball, green ball, what is going on in your mind, no one would know. Right? That is what is pretty much in a way is called hidden layer. And you can, one, classify the balls, or you can come with one strategy. If you come up with one strategy, it's called machine learning. If you come with more than one strategy, it's called deep learning in a very simplistic version. More or less, the machine also works in the similar way. For example, each of these layers can have multiple methods. For example, this node can represent logistic regression. This can be SVM. This can be other methods which come in. Basically, people don't use SVM here, but I'm just saying for your understanding purpose. And then you can switch, and then you can add different. It's called activation function. That is what is the different methods that we use to find. And one method, whatever finds, can pass that information to another method, and so on and so forth. And then that refining and refining and refining further can give you much more power to understand and classify these balls or whatever problems that you want to do. And again, when you do the deep neural network, it can be a linear, one layer of linearity, one layer of non-linearity to capture a complete chaotic non-linear one can also be done. And that's another way of doing this. So it's up to you, and it's just growing and growing. It is a fun sometimes, and obviously there are multiple examples. Like maybe less than 50 here, but sometimes we have thousands of samples like this, which with a different number of variability. So principal components analysis comes in handy. Which basically provides an opportunity to see how they are separated from each other. This is not a clustering method, but you can cluster them using the principal component. It provides something called principal components, which basically are similar to the hidden layer here, but is one hidden layer where it provides this multiple option, because still it is a machine learning approach, not a deep learning method. And you can have hundreds of principal components, but not all of them are going to capture the enough information of variability. Most of the time, the principal component 1 and 2 will have that variability, and usually people plot, but we actually go beyond the principal components 1 and 2 to capture them. And as you can see, obviously, the green and the orange ones, which are technically, they are different from each other. And this is one of my postdocs, Sam Postdoc, he did a probabilistic principal component. I don't want to go into the technique for now, but feel free to contact me if you need if you have an interest and you wanted to know more about it. But basically, when someone, this is confidence interval, probably you must have heard in simple statistics, and that's how it just kind of works. Anything that touches this red line, here are the principal components 1 to 9 that we have captured here, and anything that touches this red line represents that there is a batch associated with this. For example, the principal component 1 and 2 is what the method tries to say. Whereas if you look at the normal versus the tumor effect, again, the normal and tumor is also present in the principal component 1 and 2 and a few others as well. Now, we wanted to bring this line very close to or touching this red line by doing some statistical or machine learning analysis. And that way the batch effect would go away, and the data gets adjusted, but still this should not be removed. So that is what the tricky scenario here. And you can see here that we did it with the explore batch, and now this